Hey everyone, welcome back to biology. So we are continuing our study of our animals with now our bilaterian clades. So if you remember that term bilateral, these are animals with bilateral symmetry. So they have a top and a bottom, a front and a back, a left and a right. And triploblastic development. So all three layers, all three germ layers arise during gastrulation: mesoderm, endoderm, and ectoderm. Most bilaterians have a coelom. If you don't remember that, please go back and look that up. And a digestive tract with two openings, a mouth and an anus that are separate from each other. In our bilaterian clades, we have three more specific clades: our Lophotrochozoans, our ecdysozoans, and our deuterostomia. We described each of these clades in our chapter 32 discussion we had. So if you don't remember that, it was the very last part of chapter 32. You can go back and review that. We're going to go into these three clades and describe them a little bit more. In our Lophotrochozoans, we have four different phyla that we're going to look at: our Platyhelminthes, or our flatworms; our rotifers, which are rotifera; our mollusks, which are mollusca; and our annelids. We don't have annelids here. Annelids are a type of worm. In our Lophotrochozoans, especially our flatworms, you can see they're flattened dorsoventrally, so the back and belly are really close to each other. This helps put all the cells close to the water in the environment or close to the water in their gut. So there's a lot of surface area going on here. So gas exchange can occur. They're getting the oxygen that their cells need and releasing the carbon dioxide as waste. They also have something called protonephridia. So that part of this word tells you it's something related to something similar to kidneys, right? So these are kind of a preview of what a pre-form of what's going to become our kidneys. This is an excretory apparatus. It's a series of tubes that have cilia on them, and they pull fluid through the ducts and open to the outside to release that liquid. Platyhelminthes is the phyla that includes flatworms. They have bilateral symmetry, so you see you can draw a line right down the middle, and you have a left and right side. They also have a central nervous system, which is a big development, the ability to have a brain and nerves. However, they don't have a body cavity. They don't have a coelom. That's why they are flat. They don't have circulatory organs, so there's no heart or anything. Platyhelminthes includes tapeworms, planarians, and flukes. And they occur in two main groups. One are free living. So these are organisms that just kind of scavenge or are predators in water, mainly marine or freshwater environments, and also sometimes on land. And then there are some that are parasitic. And parasitic worms will live in or on other animals and draw nutrition from that animal and their nutrients in their body. The planarians have two light-sensitive eye spots here. So they're not as evolved as our eyes, but they do sense light, and so they are good for telling, if there's a shadow going overhead. Maybe there's a predator, or they're able to tell where the eyes are. They have lateral flaps for chemoception, so they can tell what kinds of chemicals are in their area. And they have a nervous system. That's kind of what's colored in yellow here, going through their body. So they have these ganglia at their cephalic end, which are nerve cells, nerve tissue, and then nerve cords, two nerve cords going down on their ventral side of their body, so down their belly side. Both left and right side of their body. And they have this branched gastrovascular cavity going down the middle of their body. You can see it branches all throughout here. This opens here to their pharynx and to their mouth. Then trematodes. So trematodes, we don't have a lot of flukes and schistosomes here in Tennessee, but these are a big medical problem in the world. Probably second only to malaria. They have a wide range of hosts. They have very complex life cycles. They can alternate between sexual and asexual stages. And a lot of them that parasitize humans spend a lot of their lives in a snail host. So They're in a host for a while, but they can't complete their life cycle in the snail. They complete their life cycle in a human. So they're in the water. The person is in the water, exposed to the trematodes, and then gets the trematode in their body and it develops the illness. Then through their droppings leave the larva for the snails to pick up again. These trematodes have proteins that can mimic the host and mimic the host's immune system, so they're able to actually survive and cause disease within that person. So snails eat algae whose growth is stimulated by nutrients found in fertilizer. So how would the contamination of irrigation water with fertilizer likely affect the occurrence of schistosomiasis? Schistosomiasis is the disease here that we're talking about that's caused by the schistosomes. We also have parasitic flatworms known as cestodes or tapeworms. So these are worms that can live within the intestines of humans and other animals. They don't have a mouth. They don't have a gastrovascular cavity on their own. Instead, what they do is they absorb the nutrients directly from their host's intestine. So if they're growing in your intestine, they're eating your food and growing themselves, leaving you without the nutrients that you need in your body. They're not eating or parasitizing your own body. They're just taking the nutrients from your food before you get it. If you look here at their kind of head end, they have a scolex that has these suckers and hooks that help attach it to the host so it doesn't get pushed out of your intestines. And then after the scolex, they have lots of little sections. Each of these sections are called a proglottid. And the only thing those proglottids are used for is reproduction. They have sex organs and fertilized eggs and just form this ribbon with lots of little proglottids that are just little baby making units. Those fertilized eggs are then able to leave the host body in the feces. So they're eliminated from the host and are able to then spread into, maybe those feces are left in water or in the soil and then spread to the next host who is exposed to that dirty water or soil. So how are tapeworms able to survive? They don't have a coelom, they don't have a mouth, they don't have a digestive system, they don't have an excretory system. How are they able to survive? How are they getting nutrients in them? How are they doing anything they need? Okay, our next phyla within our Lophotrochozoans are our rotifers or rotifera. These are tiny animals. They're so tiny they're smaller than a lot of protists. They inhabit a lot of fresh water, a lot of marine salt water, and a lot of damp soil. They are multicellular, they are animals, and they do actually have organ systems. They have an alimentary canal. This is a big development, that now they have a mouth and a tube that then leads to the anus. That's what an alimentary canal is. From the mouth, the tube all the way from the mouth to the anus. And this digestive tube is within a fluid-filled pseudocoelom. Hope you remember what a pseudocoelom is, where the mesoderm would be found in a pseudocoelom. One thing that's really cool about rotifers is that they can reproduce by parthenogenesis. This is when a female lays an egg, and that egg, without being fertilized, is able to produce offspring. There are some species of rotifers that parthenogenesis is the only way they reproduce. They don't have any males. They only have females that lay these eggs. So they're reproducing asexually and surviving without males. This one clade has survived 50 million years without rotifers. However, if you can remember back to last semester, asexual reproduction means every offspring is getting identical DNA as the parent. And for a population to be successful, it needs some genetic diversity to be able to handle different situations. So they're able to take up DNA from their environment. So what happens is the rotifers can be desiccated. That means they're dried out. They'll kind of get little cracks in their plasma membrane. Then when they rehydrate, they pull in DNA from outside of them. Just, random bits of DNA from dead organisms in the river or the lake, wherever they are. They pull up random bits of DNA, pull it into their cell, and use that foreign DNA to get more diversity within their genome. So they have more varied genes so that when the environment changes, there's more likely to be a rotifer that has a piece of DNA that can handle that changing environment so they don't go extinct. I hope that all made sense. I'm happy to talk to you about it if it doesn't. I think that crash course video from your homework also is kind of helpful talking about rotifer reproduction. Here are some rotifers here. Okay, so I will pick up in our next video. We're going to continue talking about our Lophotrochozoans with 2 more phyla, the mollusks and the annelids. So come back and we'll talk about that. And I will see you soon. Bye.